Hi everybody, it is your AP Biology teacher, Mr. Poser. Today we are back with a recap of Unit 3 Cellular Energetics and AP Biology. And as you can see on the screen, there are 7 topics, 2 of which are some of the biggest and most important topics of AP Biology, which are 3.5 and 3.6. But here they are. 3.1 is enzyme structure, 3.2 is enzyme catalysis. In my topic videos, I kind of combine 3.1 and 3.2 because they go hand in hand with each other. 3.3 is on the environmental impacts of enzyme function. 3.4 is on cellular energy. 3.5, big one, photosynthesis. 3.6, also a big one, cellular respiration. And 3.7 is on fitness. And we're going to talk about what that means because it's a little misleading in a second. All right, so I'm going to try and be brief and just kind of breeze through this, and here we go. 3.1, here's some of the main points. All living things, if you are a living thing, you have to be able to obtain and use energy in order to stay alive. You have to take in more energy than what you put out. Okay, and that's required of being a living thing. You can't really stay alive if you're not bringing in more energy than what you're using. So energy is in a constant kind of flux, okay, because energy can't really, it can be stored in a way, right, but there always needs to be a constant input of energy. And just like anything else that uses energy, okay, living things convert one form of energy to the next. Like light gets converted into chemical energy, and chemical energy gets converted into heat. And really, when you think about it, when you boil it down to its basic components, life itself is just a complicated array of chemical reactions. Okay, you watching this video, you digesting your food, you using this video to I don't know, keep your muscles upright or use your brain and comprehend what I'm saying. It's all just chemical reactions that happen. So really a deep life lesson there. It's just life is just chemical reactions, dude. And we can categorize chemical reactions into 2 groups. Catabolic reactions break down a larger molecule into a smaller molecule, like hydrolysis, for example, is a type of catabolic reaction, breaking something down from, say, your food into smaller pieces, like breaking down polysaccharides into monosaccharides. And anabolic reactions build up large molecules into smaller ones, like, for example, what we talked about in Unit 1, dehydration synthesis. So what is this called? This is called your metabolism, and we're going to talk about that a little bit more in topic 3.4. Okay, so since life itself is a very, very complicated array of chemical reactions, and chemical reactions are required to convert one form of energy to the next, reactions are super duper important, and living things have to make sure that their reactions are happening in the ways that they want to, when they want to, and at a faster rate, and that's where enzymes come in. So I put up here, reactions don't happen as fast as living things need them to. Living things are dependent on enzymes to mediate reactions. So topic 3.1, 3.2, 3.3 are all about enzymes. And what is an enzyme? An enzyme is a biological catalyst that speeds up reactions by lowering the energy required to start a reaction, also known as the activation energy. So something that you might see on an AP exam is this kind of graph here that shows what enzymes do. On the y-axis is energy, on the x-axis is the progress of the reaction, and what you can see is that an enzyme lowers the amount of energy required to go from reactants to products. There's a much higher slope here on this first graph, and a lot more energy is required to go from reactants to products, but when you use an enzyme, it's a lot less energy, and thus that speeds reactions up. Metabolic reactions, so all the catabolic and anabolic reactions, the ones that are breaking down and building up, are enzyme mediated. They need enzymes in order for them to function properly. And without them, well, then you're not gonna be taking in nutrients, you're not gonna be metabolizing, you're not gonna be eliminating waste, and therefore you're not gonna be alive. The maintenance of your enzymes is super important. So they are all enzyme mediated. At the same time, enzymes are highly substrate specific, and this is important to know. They only bind to and cause the reaction of one substrate. Something that we talk about a lot in my class is the enzyme lactase. If you have lactase in your, I think it's in your small intestine, then you will be able to break down lactose, which is the sugar found in dairy products and milk. You'll be able to break that down into galactose and glucose and actually use that lactose rather than letting it ferment and then make you all farty and bloaty and stuff. Basically, you're lactose intolerant if you don't have lactase. And so lactase only works with lactose. That's the point I'm trying to make. And every enzyme only works with one specific substrate. On that note, 3.2, some enzymes undergo what is called induced fit to hold the substrate in place. The enzyme changes shape to hold the substrate in the active site. So here is a really, really basic enzymatic reaction here. Here are the substrates. Those are the molecules that are going to bind to the enzyme. Those are the products of the reaction. They bind to the enzyme at what's called the active site. And when the enzyme lowers the activation energy, what it does, it converts the substrates into a product. And then it releases the product, and the enzyme can be reused for more reactions to bind with more substrates. And notice how the substrates only fit in, look at the kind of the shapes here, they only fit in to the active site of that enzyme. Okay, so the substrates are not going to bind to some other different kind of enzyme to make this reaction happen. They can only happen with that one particular enzyme. As I put here, enzymes are not part of the reaction, they are not consumed, and they are not a reactant or product. Okay, so when you're writing a chemical equation, you usually put the name of the enzyme over the arrow. Okay, that goes like products, arrow, reactants. You put it over the arrow, and that shows what enzyme is mediating that reaction. Enzymes allow reactions to occur spontaneously, meaning basically they happen on their own. An enzyme releases products from its active site and can be used again, as I said, for another reaction. It can bind to another substrate again. So the enzymes can be reused. They don't, quote unquote, participate in the reaction because they don't get broken down or built up. They just allow the enzyme, or excuse me, they allow the reaction to happen at a faster rate because it lowers the amount of energy needed, the activation energy required to make that reaction happen. Okay? On to 3.3, environmental factors greatly affect how reactions occur in a living thing and how enzymes function. All right, so a lot of how your body maintains homeostasis, which is a topic that we're going to get into in Unit 4, is making sure your body is in the right conditions to do these chemical reactions and that your enzymes are properly taken care of, they're in the proper environment, and they're going to work at their optimal pH, they're going to work at their optimal temperature, at their optimal substrate or enzyme concentration, a whole bunch of stuff to make sure that your metabolism is functioning properly. Important thing to note that enzymes are proteins. And the way enzymes are shaped has a huge effect on their ability to function. Right? So we talked about in Unit 1 how proteins have to have the right amino acids in the right order at the right time, place, whatever, in order for them to work properly. Enzymes are no different, right? So if an enzyme is slightly mutated, there's one amino acid off, the active site, for example, might not have the same shape, and it might be a completely dysfunctional enzyme because it can't bind to the substrate properly, or the substrate that it's supposed to be reacting pretty much. Okay? Enzymes all have, as I mentioned before, an optimal pH and a temperature that they work best in. Generally, higher temperature equals a higher reaction rate. So this is one factor that will affect enzyme function is temperature. So why do you have to maintain 98.6 degrees Fahrenheit or 37 degrees Celsius body temperature all the time? Well, it's because your enzymes need that temperature in order to work properly at the rate that they're supposed to. And generally speaking, when we're talking about just raw, like regular chemistry, a higher temperature results in a higher reaction or a faster reaction. And that's true for biology, but remember what we talked about Unit 1, too high of a temperature or too low of a pH can cause enzymes to denature. Okay, so breaking some of the hydrogen bonds and the side chain interactions of the tertiary structure of a protein can cause it to just kind of break apart, unfold, and then it doesn't work at all. And that's what's called denaturation. So too high of a temperature to make these enzymes denature, that's obviously not going to increase reaction rate. That is going to decrease reaction rate by a lot. It's going to make it basically the enzyme completely dysfunctional. No reactions can occur when an enzyme is denatured. Okay, so temperature up to a point can increase reaction rate. Some enzymes produce higher reaction rates at different pH. Okay, so pH is another factor. For example, pepsin is a group of enzymes that stay in your stomach, and they break down proteins. This stomach acid makes the pH very low in your stomach. So, so pepsin only works at about a pH of 2. Pepsin would not function in somewhere like the enzymes in your spit or the enzymes in your intestines, where the pH is a neutral 7 to 8-ish. And some enzymes wouldn't work, and like amylase, for example, this is an enzyme in your spit that starts to break down carbohydrates as soon as food enters your oral cavity. that works at a pH of 7, neutral pH. All right, that wouldn't work in a pH of 2. Okay, so this graph is showing, you might see again here, like, oh, this enzyme, pepsin, is going to work best at a low pH. It's going to have the highest activity at a low pH. This one's going to be right in the middle at neutral, and then this one, whatever this green one is going to have optimal enzyme activity at a high pH or a very basic environment. Okay, increasing substrate reaction increases reaction rate. Okay, if there's more substrates with enzymes to react to, then reaction rate is going to increase up to a point. It's not going to go infinitely faster the more you add substrate. Increasing enzyme concentration also increases reaction rate. Therefore, if you have more enzymes to make something react faster, you add more enzymes to it, it's going to go even faster. But again, reaction rate is limited. Okay, it's called a Vmax. You don't really need to know that unless you're in college chemistry, or biochemistry, I should say. But Vmax is basically how fast the maximum rate of reaction. Okay, and you can't just go forever. So here's why: too much substrate, not enough enzyme, or too much enzyme, not enough substrate, decreases reaction rate, not enough active sites or empty active sites. Okay, so remember, substrates need to bind to the active sites of the enzyme. And it's very substrate specific, right? So increasing either one is going to, increase the overall reaction rate. Hey, but you can't go infinitely fast. You can't go much faster than what's called, or you can't go any faster than what's called Vmax. So yeah, inhibitors, another thing that controls enzyme function. Inhibitors are molecules that control enzymatic reactions. They're basically going to stop a reaction from happening, stop an enzyme from working. Okay, so if I decide to go vegan, right, and I'm never consuming any lactase ever again, the chances are that any lactase enzymes that my body still produces, the lactase genes that are still expressed, they're going to be inhibited, right, because I'm not using it for anything. So competitive inhibitors, there's two kinds of inhibitors. Competitive ones, enzymes, and they block the substrate from binding, kind of like enzyme A over here. And non-competitive, aka allosteric inhibitors, bind at another spot of the enzyme, and they change the shape of the active site so the substrate can't fit. Okay, so here's my inhibitor. It's an allosteric site or a separate binding site, and it changes the shape of the active site so the substrate cannot react anymore, can't fit in, and it can't cause a reaction anymore. Okay, so those are competitive and non-competitive, aka allosteric inhibitors. Just recapping, here are the factors that affect enzyme function and reaction rate. Temperature, too high, too low, not good. pH, too high, too low, not good depending on the enzyme. Inhibitors can stop reaction rates from happening. Substrate concentration, how much of the substrate you put in to a reaction, that will increase reaction rate, and enzyme concentration will increase reaction rate as well. Okay, let's move on to 3.4. All life, as I said before, requires constant inputs of energy. Energy cannot be recycled, created, or destroyed. Life follows the laws of thermodynamics. Again, I said this in my other video, but you don't really need to know a whole lot about thermodynamics. All you have to know is that life converts energy from one form to the next, and it uses it, and it's just constantly kind of flowing through. All right, a lot of the energy life uses is lost as heat, so living things must be efficient with their energy. Okay, you cannot be losing more energy than what you gain, otherwise you're going to die, pretty much, if you're a living thing. Doesn't matter what kind of living thing you are. You have to follow that rule, or it's over for you. Sorry. This is a metabolic map. I'll talk more about that in a second. Metabolism, as I kind of alluded to in 3.1, is the totality of all of an organism's reactions. Anabolic reactions build larger from smaller ones. Catabolic reactions break down larger molecules into smaller ones. We talked about this already. All of those together are called metabolism. And metabolism can be diagrammed from metabolic pathways, sequences of reactions that transfer energy. So how is this energy conversation going into the reaction conversation? Well, I'll show you right now. Here's a simple metabolic pathway. This is the same thing I did in my other video. Let's just say A is converted to B, or let's go, molecule A is converted to molecule B is converted to molecule C, and molecule C is converted to molecule D. To be efficient and not to needlessly waste energy, because you need a constant positive gain of energy, living things use energy or reaction coupling, using the energy released from one reaction to power another reaction. So it's really kind of simple. If one reaction releases energy or causes a net output of energy, it's called exergonic. And then if a reaction causes a net loss of energy, or it requires more energy than what it's going to release, there's a net loss of energy. It's called endergonic. And simply put, exergonic reactions, those that release energy, These are used to power the endergonic reactions. So the living thing is definitely using this energy to the most extent possible. It's not just letting the extra energy from the exergonic reaction dissipate into the environment as heat. It is being used, and therefore it's being more efficient. And this is called energy coupling or reaction coupling. And as I said, exergonic reactions power endergonic reactions through energy coupling. Okay, so this is pretty straightforward here. I wanted to talk about this metabolic map. Okay, I showed you one simple metabolic pathway. All right, if you combine a whole lot of metabolic pathways in any kind of living thing, you can build a whole map of metabolism here. And this is a very, very brief example. This is brief, I'm telling you. Brief example of metabolism here. So like, check it out. Here's anaerobic sugar respiration. Here's, let's see, where is, see, I can't even find anything. Citric acid cycle, there's the Krebs cycle, right? We're about to talk about that. Purine biosynthesis, yeah, a whole bunch of stuff, all right? But you can build a map, right? And you can study that pretty extensively. Now, to talk about some of the main reactions, the most important sets of reactions in living things, we're going into photosynthesis and respiration. 3.5 is on photosynthesis. And what is that? It is the process by which living things capture light energy and convert it into chemical energy stored in organic molecules. This is miraculous. All right, when this evolved, it changed life on Earth forever. Living things have an ability to take a non-living energy source like light and convert it into a form that living things themselves and other living things can use. That is miraculous, and that cannot be understated. I know I sound like such a nerd right now, but I am. All right, I'm owning it. What photosynthesis does, it provides usable energy for most of life on Earth, and it produces oxygen as a byproduct. And we're going to talk about why that's super important later on. Super simple diagram of photosynthesis right there. Real quick about photosynthesis, I just said, like, when this evolved, it changed Earth forever. Yes, these organisms called cyanobacteria, they're still around today. First of all, the ability to photosynthesize about 3 billion years ago. And cyanobacteria still produce about 80% of the oxygen in the atmosphere. So the oxygen that you're breathing right now is from the ocean, is from cyanobacteria. So we're all like, oh yeah, thank a tree, hug a tree for letting you breathe and stuff. But it's like cyanobacteria. It's little tiny green guys that are going to produce more oxygen than all the rainforest combined. Yeah, modern chloroplasts evolved through endosymbiosis of cyanobacteria. That's what we talked about last unit. Photosynthesis itself happens in two parts, the light reactions and the Calvin cycle. And here's a really brief diagram. I'm going to talk about these momentarily. It's a really nice representation of how this works. Okay, the light reactions is phase one. Light reactions are called the light reactions because they involve light. This is the photo part of photosynthesis. Photo means light. And they occur in the thylakoids of the chloroplast. So we're talking about the anatomy of a chloroplast last unit, and this is why. Light is absorbed by chlorophyll. It excites an electron, which is then transferred between proteins to produce ATP and NADPH. That is the overall picture of the light reactions. And so here's a thylakoid, right? Remember the little discs, the Oreo-looking things, or Girl Scout cookie-looking things in the chloroplast? Here's one of them, right? Along the membrane are two photosystems, photosystem 2 and photosystem 1. And an electron transport chain occurs along what's called the thylakoid membrane. Here's the stroma, here's the thylakoid lumen, the inside of the thylakoid. Light is absorbed by one of these. It excites an electron, starts a chain, and then provides the right environment to produce some ATP through what's called a proton gradient, and it produces NADPH. Okay, so here's the basic steps here. I'm going to try and go through these as quick as I can, but as effectively as I can. Number one, a water molecule. Where does it get the electron from to pass that around from one protein to the next? It comes from water. That's why plants need water. They need the electron from water. And they split that into hydrogen ions and oxygen gas. So it'll take two water molecules to do that, right? Because O2, right? So yeah, it gets the electron from water. And water is the main electron donor. Step number two, photosystem 2 is a protein complex involving chlorophyll and a couple other proteins. I don't remember the name of them, but its job is to excite the electron using light energy. All right, so the electron kind of absorbs a photon of light. Do we have to know the specifics of how that works? No, I couldn't really explain it to you if I tried, but basically the electron absorbs the light energy, and then that electron gets passed from one protein to the next. It gets passed from photosystem 2 to photosystem 1 using an electron transport chain. And now, using that energy in that electron transport chain, it establishes what's called a proton gradient. It's a proton pump. Remember those from last unit too? actively transporting, without ATP this time, but actively transporting hydrogen ions from one side of the membrane to the next. It's going to pump in some hydrogen ions into the thylakoid. It's going to establishing a proton gradient between the stroma and the thylakoid. And then another photon of light takes that electron and re-excites it in photosystem 1, and then that electron is passed around, and this time, instead of establishing a proton gradient, this electron is now going to help catalyze the reaction of NADPH, the formation of NADPH. And NADPH is an electron-carrying molecule. It's a reducing agent. We're going to talk about NADPH and NADH here momentarily. All right, and step 7, ATP synthase, makes ATP using a proton gradient. We're going to talk more about ATP synthase and how this works, but basically this is an amazing enzyme that what it's able to do is use a proton motive force to phosphorylate ATP. Okay, so ADP stands for diphosphate, ATP stands for triphosphate. It's able to use the motion, the actual physical motion of those protons from one side to the next, okay, because we pumped them in, they want to get back out, right? That's a concentration gradient. They want to get back out, and the way for them to get back out is through ATP synthase. So ATP synthase takes the actual motion of those protons and uses that energy to phosphorylate, add a phosphate to ADP to make ATP. I'm going to talk more about that in a sec. Okay, as I said before, NADPH is a reducing agent, which means it carries electrons and transfers them. That's basically its job. It carries electrons. ATP is the energy currency molecule. We're going to talk more about that in a sec. Okay, and those two molecules are used in step 2 of photosynthesis, which is called the Calvin cycle. We don't have to know a lot of the specifics about the Calvin cycle. Okay, but the Calvin cycle, what you do need to know, it takes place in the stroma. Okay, so outside of the thylakoids, in the chloroplast. It involves fixing carbon dioxide, so absorbing basically carbon dioxide from the air and fixing it and putting it into sugar molecules using energy from the ATP and the NADPH from the light reactions. Okay, so the Calvin cycle cannot happen without the ATP and the NADPH from the light reactions. So basically, I know this Calvin cycle is like, oh yeah, these are the dark reactions. It doesn't need light to do this. It does. Okay, if you shut a plant in the dark, it's going to die. So yeah, okay, the light reactions, the products of those are needed to convert carbon dioxide into glucose during the Calvin cycle. All right, here we go. 3.6, cellular respiration, the process by which cells produce ATP from macromolecules. Big idea. There's two main types, fermentation and aerobic respiration, and this is the overall chemical formula of cellular respiration. Okay, I should say aerobic respiration. Every cell takes what you eat in the form of glucose, combines it with what you breathe into oxygen, makes carbon dioxide, which you breathe out, it makes them water, but most importantly, it makes ATP. And ATP is ultra important. It is the energy currency molecule. You can't live without it. As soon as you run out of ATP, you're dead. Okay? Your cells can't really do anything. They can't do active transport. They can't do a whole bunch of stuff. Fermentation is the degradation of organic fuel without the use of oxygen. What I just walked you through is aerobic respiration. Okay, but ATP can be made without oxygen. That's called fermentation, aka anaerobic respiration. It is less efficient, meaning it produces less ATP than aerobic respiration, and that's done by most prokaryotes and a few eukaryotes like yeast. Okay, so prokaryotes don't have a mitochondrion, right? They don't have membrane-bound organelles. They have to rely on stuff like fermentation in order to make their ATP, and they can do that, right? Glucose is converted to pyruvate, and pyruvate is thus converted into lactate. Okay, but the process of producing glucose to pyruvate nets 2 ATP molecules. So it's like, oh, cool, we can get 2 ATP molecules from a glucose molecule. That's the point, right? We want to use that carbohydrate to get energy. Goody, right? But here's aerobic respiration. That produces ATP using oxygen. It's more efficient, and it's done by eukaryotes because they have the mitochondria to do that, or in some prokaryotes. But the goal of cellular respiration, is, as I was saying before, is the phosphorylation of ADP into ATP. So here's ADP, right, and what we call an inorganic phosphate. ADP, here's adenosine, all right, it's got a pentose sugar. It's actually a derivative of a nucleotide, so it's kind of like adenine a little bit. It's got 2 phosphate groups right here. That's ADP, all right? And what we're trying to do through cellular respiration is provide enough energy to add this 3rd phosphate to ADP to make ATP. And ATP can be used by cells to do whatever they want. Okay, they can use ATP. They can control how they use ATP and how they use that energy. So that's our goal here, to make ADP into ATP, and that's called phosphorylation. And as I put down here, it is the cellular energy currency for most cellular processes. You really can't do anything without ATP. Super important stuff. Okay, active transport being one. Aerobic respiration takes place in 3 stages: glycolysis, the Krebs cycle, and what we call oxidative phosphorylation, or the electron transport chain. Here's glycolysis. I pointed this out before. This is step 1. Everybody does glycolysis. Every single living thing does glycolysis. Doesn't matter if it's anaerobic or aerobic. The anaerobic breakdown of glucose into 2 molecules of pyruvate. It's a series of reactions that produces 2 ATP. Yes. 2 NADH. Yes. But it also produces lactic acid, or lactate, as a byproduct. If you're a yeast cell, okay, you're going to produce ethanol as a byproduct, and that's how you like brew beer and and alcohol and that kind of stuff. But that's a topic for another day. All right, but lactic acid is a byproduct of glycolysis, anaerobic respiration. So you're working out, right? If you, let's say you're going for a run and you haven't worked out for a while and you go into oxygen debt, your muscles are not getting enough oxygen. They're going to resort to using this kind of respiration rather than aerobic respiration. They're going to be producing less ATP, and they're going to produce the lactic acid that makes you cramp. So stay in shape. NADH is an electron-carrying molecule, aka a reducing agent, very similar to NADPH, as we talked about before. And glycolysis occurs in the cytosol, not in the mitochondrion. In order to get it into the mitochondrion, pyruvate, okay, which is the end product of glycolysis, is converted to a molecule called acetyl-CoA and transported into the mitochondrial matrix, the very inside of the mitochondrion, which is where the Krebs cycle occurs, and that's stage 2. And as I put here, that is a cycle of reactions that uses acetyl-CoA to produce 6 NADH and 2 FADH2, another reducing agent molecules, as well as carbon dioxide, into more ATP. Hooray! More ATP, more NADH, more FADH2. Yes, we're on it. We're on it. Okay, 2 ATPs from one process, 2 ATPs from the other. We are cooking with gas. No, we're not. Not really. I'm going to show you here in a sec. But this is step 2. All right, this is aerobic. Oxygen is required in order to get to this part here. Oxygen and mitochondria are required. All of the NADH and FADH2 produced through stages 1 and 2 are used in stage 3, which is oxidative phosphorylation, or the electron transport chain. Okay, so right up here, okay, we said, oh yeah, we produce some ATP. We produce more ATP over here, like 2 each. Nice. Okay, we can produce anywhere from 30 to 36 ATPs using 1 glucose molecule from the beginning of this step process at maximum efficiency. That is huge. So way more ATP this time than in step 1 and step 2. Okay? And how exactly does that happen? Now, how does that even make sense? If we look at our formula from a couple slides ago, it's like, okay, how do you get C6H12O6 into 36 ATP? This is how. All right, and just before we get into that, here's our 3 stages, and this is where they happen. This is the cytosol, glycolysis. Okay, it's converted to acetyl-CoA, the mitochondrial matrix. The very inside is where the Krebs cycle occurs, and then along the inner membrane, the convoluted, folded, crysty inner membrane of the mitochondria is where the electron transport chain, oxidative phosphorylation, occurs. And so that's what I just said. ATP synthase is powered by a proton motive force to phosphorylate ATP. So I mentioned ATP synthase before, right? And now we are just basically, guys, what we are doing in oxidative phosphorylation is we are cranking the crap out of a proton gradient. We're creating a very, very strong proton gradient, so ATP synthase just goes into hyperdrive. It just goes, it actually spins like this. It's kind of cool. It's just using a huge proton gradient, and we are producing ATP like unbelievably quick. All right. So how does this even work? Okay, I'm going to show you. NADH and FADH2 donate electrons to proteins called complex 1, complex 2. Excuse me. NADH to complex 1, FADH2 to complex 2. They're dropping off electrons, and they're starting, once again, an electron transport chain. Check it out. Complex 3 and complex 4 are, check it out, once again, they are proton pumps, and they are producing a big old proton gradient for ATP synthase to use. Electrons are transported from 1 to Q to 3 to cytochrome C and to 4. That's the name of these proteins here. I don't think you have to memorize those for the AP exam, but you have to know how this works for sure. Yeah, so those are electron donors. Like I said before, that's what reducing agents are. And 3 and 4 and 1, 2, hey, they are producing a proton gradient. They're pumping hydrogen ions using those electrons, using Yeah, using the electron's energy into intermembrane space, creating an electrochemical gradient, a proton gradient. So we have a boatload of hydrogen ions in the intermembrane space. Where do they want to go? Okay, well, they want to diffuse, right? They want to go from where there's a lot of them to where there's less of them, and the way for them to do that is through ATP synthase. And ATP synthase shoots these basically protons back in the way they want to go, and it spins. It actually spins. It goes, okay, due to that proton motive force. And those protons are going to move back into the mitochondrial matrix, and they're going to be able to phosphorylate ADP into ATP using the motion of those protons. It's called a proton motive force. It's actually fabulous. It's amazing, and it's able to make anywhere from like 30 to 36 ATP molecules just using that. Okay, it's able to phosphorylate ADP into ATP like 36 times. Okay, where does the electron go that gets bounced around though? The last electron, the last place the electron goes is to an oxygen molecule. Oxygen, why do you need to breathe? To accept electrons at the end of the electron transport chain. That's why you need oxygen. Okay, so I've asked kids before, like, why do you need oxygen? They're like, to breathe. Why do you need to breathe? To live, right? But like, this is actually what you use oxygen for. It is a terminal electron acceptor. So that electron is not bouncing around wreaking havoc on your mitochondria. The electron gets accepted by oxygen, and it turns basically into water. Okay, so it infuses with that electron, and it fuses with these hydrogen ions, and it turns back into a water molecule. Okay, so we've gone full circle. Water during the light reactions donated an electron. Oxygen is accepting the electron and getting converted back to water. And water is the byproduct of this whole thing. All right, last topic. It's pretty quick. 3.7. Is talking about how organisms have a wide variety of molecules. Why? Number one is enzymes and receptors only work with one other molecule. We already talked about this. Enzyme substrate specificity. Okay, but organisms have a ton of different types of molecules. Hey, and this is one reason, but the main reason why is because having a large molecular diversity enables organisms to respond better to environmental stimuli. Okay, an organism is more adaptable to its environment if it has a large variety of enzymes, it has a large variety of structural proteins, receptors, whatever. A huge variety of molecules makes an organism more fit and more adaptable to many environments, and that's what we call fitness. This is a huge, huge, huge idea in biology. More variety equals better survival and reproduction. This is going to be a concept that we come back to again and again throughout this class. Okay? Whew. All right. I believe that is it. Yep, that is it. Let me know if you have any questions. I did my best to be brief here, but if you have any questions, let me know. Have a great day.